Good morning, good afternoon, good evening, wherever you are. Very happy to be here and talk to you about our research we do in the past 18 months. And since, as you see, I'm a veterinarian, I work in the realm, in the interface of animal and human, and animals and humans. So in the One Health research approach. And I'm a professor at Kansas State. It's in the middle of in the country. It's actually in the middle of nowhere, so to speak. It's in Kansas. And we have a really nice veterinary school, about 110 veterinary students a year. We are 2 hours west of Kansas City. Many people have heard about Kansas City, 2 hours west and 7 hours east of Denver. So it's a good place to do what I do, namely infectious diseases, and we will go through that in a couple minutes. So before I start, I have to acknowledge my collaborators. And the work I do takes many people, as you see. It takes a village. Without the people in my lab, they're listed here on the left. I have molecular biologists, I have pathologists, I have technical support, I have people who do clinical pathology, I have people who do immunology. Without these people, and they have worked incredibly hard for the last 18 months, I couldn't present what I present today. We were shut down on March 15th at the university for every research except SARS-CoV-2. And we actually did not close my lab and my offices for 1 day because we just continued what we had done before with SARS-CoV-2. And then I have to mention my collaborators. So this is my lab, my collaborators, Wenchung Ma, who was with me at Kansas State for many, many years, and he moved to Missouri 6-8 months ago. My long-term collaborator, Bill Wilson from USDA, Upper which is also in Manhattan, Kansas. I'm from the Little Apple, Manhattan, Kansas. And then we talk about therapeutics at the end. I work with Sumit Chanda and his team at Sanford Burnham Brabis out west there in Scripps area in La Jolla. And also on therapeutics with Adolfo Garcia Sastre and Ivan Marazzi from Mount Sinai School of Medicine, New York City, and pathology I do with my good colleagues from Louisiana State, Udeni Balasuria and Mariano Carosino. And without money, we cannot do anything. So I'm very grateful for the funding. We can talk all day long, but if we don't have money, we cannot do anything. And I got funding from the NIH, National Institute of Health, from the Department of Homeland Security, from USDA. I got some money from the state of Kansas and BALF transition funds. And at the end of last year, I got money from the WHO. And this research I do is very, very expensive. One experiment, which I show you soon, will cost about $150,000 to $200,000 with these animals in high containment. So anyhow, the outline of my presentation, you have heard that, I believe, in the last couple of days several times. Concept of One Health. I go fast through that, but I want to maybe add some points which I believe are critical. I introduced SARS-CoV-2. We have heard about that many, many, many times, but I give you one slide overview. And then we talk about my centers. And as I was told, as you can read in my CV, I'm the director of 2 national centers of excellence. One is CEZAD, and the other one is CEZID. One is from the Department of Homeland Security, the other one from from NIH. And then we talk about studies we have done in livestock, in pigs. We also did companion animals, and I tell you why. And then we did deer. That's a study not published. The first 2 are published. And at the end, I will talk to you about our therapeutic approaches to COVID-19. We have all these beautiful vaccines now out there, and I encourage everyone to get vaccinated, at least when you are at the age that you're able to get vaccinated. Get your vaccine and get your booster shot because they are safe and efficacious, and that's the only way we can conquer this pandemic. So anyhow, Concept of One Health. Dr. Rudolf Virchow, a medical doctor who lived in the early 19th century, a very famous pathologist, actually, from Germany originally, Berlin. He said, between animal and human medicine, there is no dividing line, nor should there be. The object, animal versus human, both are animals at the end, is different, but the experience obtained constitutes the basis of all medicine. And that was the basic, the concept of One Health. And One Health, as we all know, unites the spectrum of medical and ecological experience with the goal of improving and protecting the health of humans and other animals, plants, and our environment globally. And this is how you have seen that many times. Everybody does it different: animal, plant, ecosystem, wildlife, human health. Together is One Health. And you have heard that most likely many times, that about 70 to 75% of emerging infectious diseases in humans are zoonotic in nature. And most of them come directly from wildlife and jump into humans. Sometimes they have an intermediate host, and we discuss that in a minute when we talk about SARS-CoV-2. And so, for example, highly pathogenic avian flu H5N1 directly into humans. There's there's from chickens. If you buy chickens which are infected, and you get infected by eating them or by slaughtering, etc., and cutting them. And we believe, and I believe, that important is biosurveillance. And this needs the integration of both human and animal populations worldwide. And I'm a veterinarian, as I told you in the beginning, and I don't teach much, but when I teach, it's very important that we teach veterinarians a lot about these emerging zoonotic diseases, because the veterinarian is the only health professional who likely sees both the animal owner and the animal. And if there is a disease which is similar in animals and humans, hopefully the veterinarian can put one and one together. The human doctor, medical doctor, only sees the human patient, but not the animal patient. We, as veterinarians, see both, and that's why it's very critical to educate not only the young veterinarians, but also the people in the field, to educate them about zoonotic diseases. And what are our challenges? And this is a slide I borrowed from Dr. Fauci. He published that 15 years ago already with others, Tony Fauci, and he already talked about emerging and re-emerging diseases. And you see so many diseases here. SARS-CoV-2, first described in 2003, H5N1 influenza in 1997, Nipah Hendra viruses in the 90s, human monkeypox, Ebola, we know for many years in Marburg and in parts of Africa, and in the United States, various anthrax. We had the bioterrorism in 2001, we had importation of monkeypox and other things. So there are many of these diseases, and now we cannot forget the multi-drug resistant tuberculosis and malaria. There are many diseases which emerge for the first time or re-emerge. Another one I forgot to mention is West Nile, which arrived in the East Coast in New York in 1999 and made its way all the way to the West Coast in the meanwhile. And now in the last 10 plus years, here I have since 2009, we have many more. We have MERS coronavirus in 2012. We have SARS coronavirus 2, 2019. H7 and 9 influenza, a different subtype, first time described 2013, a big problem. I'm really concerned about that. Ebola, the big outbreak in West Africa, 2014-16. Zika, you remember, coming in from South America, actually into Florida, even in 2015-16. And then we had pandemic H1N1, the last pandemic in 2009, most likely originated in swine in Mexico. And then we had an outbreak of highly pathogenic avian flu in 2014, mainly in Midwestern states, Iowa and other states, and it cost the United States $4 billion to get to eradicate that disease. So this is a emerging theme. There are always new diseases coming, and many of these all of these are kind of zoonotic. And why are these not only important for human health? Some of these diseases I work on, and we don't go through that today, are transboundary animal diseases. They affect animals like pigs or cattle, etc., and they have incredible economic impact. And this is just a summary. What is the economic impact of some of these diseases until about 2010? BSE, you remember, bovine mad cow disease in the UK, that cost $10 to $13 billion. That was in the 80s. Not much compared to the outbreak of foot and mouth disease, which is a viral disease of cattle, sheep, and pigs, ruminants and pigs, which cost in the UK, they had 2 outbreaks in early 2030, $30 billion. The SARS-CoV-1, the first SARS outbreak, 2003, mainly in China, Hong Kong, Singapore, and also was exported to Toronto, $45 billion. The avian flu, the expansion of H5N1 from China, Hong Kong, all the way to Central Europe and all the way to Canada and America, $40 billion. And that was before we had it in North America, that was $40 billion. In the last 10 years, pandemic H1N1, $300 billion. Ebola, West Africa, $53 billion. And recently with SARS-CoV-2, the estimates are 20 plus trillion in the next 4 years. Worldwide impact, economic impact. And African swine fever, it's a disease I work on, which made its way into China in 2018 and all over the Southeast Asia, had already an economic impact of 100 billion. And this is not a zoonotic disease, only affects pigs, not people. And this virus wiped out about 25% of the world's pig population within a year. Incredible. So what are our challenges? So we have the zoonotic diseases, we have these animals, severe animal diseases, which are spreading and emerging in different parts of the world. Our challenges are that we have to feed the world. And right now we have about 8 billion people on this planet, and in 20, 30 years, we have 10 billion. So we have to feed these 10 billion people. And that's a challenge if you rely on food animals and they have diseases. And for example, in 2018, worldwide, we had 4 billion production animals. And we need, in order to feed 10 billion people, we need 50% increase in production animal production. And this is a summary which I borrowed from the FAO. And here they showed that from 1960 and within 50 years to 2007, the increase in production of chickens, of poultry, was 10 times, from 7 million tons to 75. And eggs was 4 times the production increase. Beef, 2.5, and pork 4 times. So incredible increases in poultry and pork and also eggs. Beef is not so significant. It's significant, but not as much. And it's predicted that in the next 10 years, we have to produce about the same, double our poultry production in order to feed the people in the world, to 50% more egg production, 50% more beef production, and 25% more pork production. So these are the challenges if you deal with diseases which have either infect only animals or have also a zoonotic effect. So they are circulating in animals and transmit to humans, like swine influenza or avian flu I mentioned. So what was our response when we had, in March of this year, we had this new pandemic coming, rising, emerging around the globe. we started to work on SARS-CoV-2, and the rationale and why we can do that is at Kansas State, we have a facility which is called the Biosecurity Research Institute. And this is a high containment facility. High containment means we can work with agents which are highly virulent and dangerous for human health in a safe environment. We work with these agents, biosafe and biosecure, and we work in BSL-3 and BSL-3-EC facilities. We have animal rooms, which we have 5 animal rooms where I can hold about 32 cattle in BSL-3-EC environment. And then we have several laboratories where we can work with these highly infectious and dangerous agents. And that's why we started right away. We already, before the shutdown happened, the lockdown of the university, we started to work with SARS-CoV-2. In my shop, because I do a lot of animal species, as you see here. We work with mice, hamsters, all the way up to horses and camels, also deer. And I work with many different diseases like Rift Valley fever, I mentioned highly pathogenic flu, swine influenza, African swine fever. They all have to be worked in very high containment conditions. And that's why I work with these. And I added 1.5 years ago also SARS-CoV-2. So what is SARS-CoV-2? It was first described in December 2019 in Wuhan, China. It emerged the first time. Was it the first time it infected a human being? I don't believe so, but it is, at least it was first described in December 2019. It's a coronavirus. I'm a virologist, so I give you a little bit. It's a coronavirus. We have in viruses, we have orders, families, genuses, and subgenuses. But what we need to know here, it's a coronavirus which belongs to a genus called Beta coronavirus genus. And it's a very long RNA virus. The longest RNA virus known in mammals, or it's 30 kilobases, means 30,000 bases long. Influenza, for example, only has 12,000. So it's very large RNA virus. RNA viruses are usually, in general, smaller than DNA viruses, in general, not all, but 30 kilobases is pretty good. African swine fever, for example, is 180, between 170 and 190 kilobases. That's the difference. And this is an RNA virus, a positive strand, non-segmented, has various genes. And one of the genes you hear all about, and I don't go in detail today, is the S, the spike protein. This is on the surface here, as shown here. And this spike protein is needed to attach to the cell receptor, which that virus recognizes, and then there are certain, and this allows the virus to invade a cell, infect the cell, and replicate or take over the machinery of the cell and replicate in the cell and produce new progeny. So the spike is here in the genome, and it's very important for virus attachment and entry. And it's attachment on the cell, and we get back to that, is it needs a receptor for that, and it's called the ACE2 receptor. You most likely have heard about it. It's angiotensin converting enzyme 2, and this is the receptor on the surface of cells it uses to dock on, and then it is allowed the cell, and then it fuses with the cellular membrane via the fusion peptide within the spike. And we also know that human-to-human transmission is very efficient, mainly via respiratory droplets, but also airborne. And we did do a lot of work, and there's also indirect contact. There's not much known about it, but we published several papers on the stability of SARS-CoV-2 in biological fluids, human biological fluids, and on surfaces under different climatic conditions. You can read that up and find that under my name. The zoonotic origin, we just touched very shortly on that. We don't know the precise origin of SARS-CoV-2. You know, there's a big debate right now, and it's becoming more and more political. And I'm asked also all the time by media outlets and journalists what I believe. The best evidence so far suggests that bats are involved, and maybe pangolins, possible. But we will, in my opinion, never know what really happened. We are 1.5 years after the fact. This is too late to go all discover or study all the different avenues this virus could have taken. So this is a summary by McLean and others just published a couple months ago, how they think it could have happened. It's a virus which started in a bat. This virus obviously transmits between bats and gets adapted, maybe recombines, and then through whatever event, it either directly was transmitted to humans from a bat, or it had maybe a potential intermediate host. And in this direct transmission from bat to humans, we cannot exclude that some bat virus was isolated in some laboratory in wherever, maybe in China, maybe in another part in Southeast Asia, and it happened in a laboratory and was not recognized early on. I would not exclude it. We have to keep all options open. So, but when we think about this normal natural transmission via bats into maybe an intermediate host, and one intermediate host is the pangolin, and here's the bat virus, the closest to the SARS-CoV-2 animal virus, this RATG13. We see that this one is closer than the pangolin virus they isolated. If you look at the very important motif, which are 5 amino acids in the receptor binding domain of the spike, and this is L, F, Q, and Y. These are the regions, the amino acids, which interact with the ACE2 receptor. You see that the pangolin is 100% identical in this region, very important region, whereas the bat virus only has 1 out of 5. So the similarity is not so good. And when you look at another hallmark of this virus, namely this furin, you might have heard about in the spike, there's a furin cleavage site, which is a series of amino acids, the basic amino acids like RR, AR. None of these motifs is not present neither in the bat nor in the pangolin virus. So I believe we will never know really what happened. We are 1.5 years too late. So anyhow, so what do we know about SARS-CoV-2 and natural infection in animals? We discussed now the bat pangolin theory, and now we know since the last 1.5 years, there have been many infections in cats, domestic including and large cats, in dogs, gorillas, in primates, and in mustelids, including mink, ferret, otters. And this is a summary here. We have discussed it in a paper already last year, why it is important to study animal populations. We know, for example, that lions, tigers, leopards, cougars, gorillas, otters have been infected. Some of these big cats demonstrated clinical signs, and most of these transmissions were through animals, through caretakers which were positive. And there was also a transmission to co-housed cats. In dogs, in a survey, they showed that 7% in a highly contaminated, human-contaminated, SARS-contaminated area had positive neutralizing antibodies. Other surveys didn't see that. And in domestic cats, even higher percentage of seropositivity, up to 15%. In mink became very interesting because many farm minks in Europe in the beginning were reported mink farms to be positive, and again positive by caretakers or owners which shed SARS-CoV-2, and they really reacted with severe disease, even mortality, and then later on it was found that mink farms in the United States and Canada were also positive, and in the United States they found one wild mink which lived very close to a mink farm. They caught it and found it to be positive. They also looked a lot in ferrets, which are other mustelids, and found that ferrets in COVID households, some of them became also positive. The most important thing is This we call the transmission from human to animal, we call reverse zoonosis, because the zoonotic event is from animal to humans. So these are reverse zoonotic events. The most important one is that these mink farms, many of these mink got positive and they transmitted it back to naive humans. And that could be traced with their genetic code. So there were mink-like viruses which were later found in humans, which interacted with these mink. So there was both reverse zoonotic and then zoonotic transmission. And obviously it's very dangerous that you have a secondary reservoir where viruses with a different genetic code, with a different genetic and maybe phenotypic properties jump back into humans. However, we have to say that these SARS-CoV-2 in animals is rather limited when you consider millions of people have been infected. So the first study we did already in April last year was we got the virus early on, and then we said, why don't we look into pigs? Because I work a lot with pigs, and pig is a very important commodity and export market for us. In the United States, we produce 100 million pigs a year and export billions of dollars of pork. And this was published last fall, so I go very fast, 2-3 slides through this. And you can read it up in this paper in Emerging Microbes and Infection. And several lines of evidence suggested that pigs are susceptible to SARS-CoV-2. The first one was that that pigs were susceptible to the first SARS-CoV, the 2003. And that was by work done by a Chinese group. They found in a piggery in China that some humans got infected from the pigs. Sorry, vice versa. That some pigs got infected from humans infected with SARS-CoV-1, we can call it. So they showed reverse zoonosis from humans to pigs. And then my colleague Hannah Weingartel in Canada showed when she infected pigs simultaneously, intravenously, intranasal, orally, and per us, and that was ocularly, with SARS-CoV-1, she could detect RNA for 1 week. These animals were shedding RNA and also developed neutralizing antibodies. And we could show very early on, before we did the pig experiments, we infected porcine kidney and testicle cells, and they were highly susceptible to SARS-CoV-2. Another line of evidence is shown here. People very early on did detailed structural analysis. This is in silico analysis, where they used, for example, the ACE2 receptor of pigs, ferrets, cats, non-human primates, and in this case for civet cat. And they showed how well these interaction could work, and when they looked in more detail, they said pigs, ferrets, and cats look very good, similar amino acids in the critical motifs or the critical amino acids as humans, and they would be very good targets, susceptibility for SARS-CoV-2. So then we did the transmission study, and I summarized that in this slide because it's published. We used 9 pigs, and we infected it with the early isolate WA1, 1 million TCID50 means 1 million infectious units, 50. And we infected not only intranasal and per os, we also infected into the trachea. We call it intratracheal, because if you have a respiratory virus and you instill it into the trachea, that's a very severe challenge. And then after a day, we add sentinels because we want to see that if pigs get infected or animals, whether they transmit it to not naive, non-infected negative animals. So we added these. So we had these 2 pens or stalls, and we had in each stall 3 sentinels and 5 or 4 principal infected animals, as shown here. And then we euthanized animals at several time points, 4 and 8, to see whether we find the distribution of the virus in different organs and maybe lesions, like in the lung. That's where we look very closely: lung, trachea, bronchi, and the nasal turbinates and the upper respiratory and lower respiratory tract, as we call it. And then we, at 21 days, we necropsy the sentinels, euthanize the sentinels, and want to see whether they have some lesions and some antibodies, because antibodies usually take 2 weeks, 2-3 weeks before they develop. And then we test for all these different markers. And our summary is here. We could not find any susceptibility. VIAD is very severe inoculation, intratracheal, intranasal, oral. We did not see any transmission to sentinels, the contact animals. They were all negative. And none of the animals seroconverted, neither the principal infected animals nor the sentinels. And so we concluded that pigs are unlikely to be significant carriers of SARS-CoV-2. We did another study with my good colleague Heinz Feldman and Vincent Münster at the Montana Rocky Mountain Lab, and they used younger pigs. We had 5-6-week-old pigs, and they used 3-week-old piglets from the same farm. So we have the same farm, and we had the same virus, and they did IN, the same inoculation plus intravenously, and they did not see any clinical signs, nothing. However, they saw some seroconversion, most likely due to intravenous inoculation of the antigen. So then we moved on to work on domestic cats, because there were so many reports. And that was the first time in my career as a veterinarian to work with a companion animal in an experimental setting. I only worked with companion animal in a clinical setting. And that was very hard to do. But we had to do it because we need to inform public health and veterinary authorities what's going on and why these animals might be important or not important. So we did our first study, that was maybe in May, susceptibility and transmission of SARS-CoV-2. And this is mainly done by Natasha. It's also published in fall. And Mariano, my pathologist at Louisiana State, and Jesse, my pathologist at Kansas State. Why cats? Because cats are very important hosts of alpha and beta coronaviruses, and they induce diseases in these animals, and they can cross species barriers. We know that. In cats, mainly, these are alpha coronaviruses, which replicate in cats. There is evidence from the SARS coronavirus epidemics, the SARS-CoV-1 in 2002, 2003, that cats are susceptible. And which in my introduction, I told you that that domestic and zoo cats, large cats, were also susceptible and infected. Sorry. And we all know that cats have close interactions with humans and could serve as a potential secondary reservoir where the virus could replicate and jump back, spill over back to humans. And we have about, in this country, in the United States, we have about 190 to 100 million house cats. And the problem here for me, I see, are the wild cats, the feral cats. And we have about 60 to 70 million feral cats in the United States only. And they can get infected because they get fed every day by people, and these people could be infected and transmit the virus to them. And so the virus could replicate and go out in the wild and so on. And it builds secondary reservoirs. And sometimes we need susceptible species like cats as preclinical animal models for study disease and development vaccines and antivirals and therapies. And as you know, there is a new vaccine now out for animals. They were tested first in cats in my laboratory, and now they're donated by the company to vaccinate zoo animals, gorillas, the wild cats and zoo animals all over the United States. So it's very important to establish these models, and we use that to do efficacy studies with that vaccine, which is now used to protect very valuable animals in zoos around the United States. So what about how do we do these studies? We used not many animals. We had 2 groups of 3 cats, as shown here, in 2 cages. And we challenged them intranasally and per os with, again, 1 million TCID 50. This is infectious dose 50 of the original Washington strain. We also add to each of these cages, shown here in dark, 2 sentinels. These are normal cats, which are added after 1 day to see whether there's transmission to sentinels. Because we have never worked with cats, we needed to control cats to do necropsies and to get the materials and organs and tissues which are negative as our negative controls. We euthanized 2 animals 4 days after infection, 2 of the principal animals, and we necropsied or euthanized another 2 of the principal infected animals at 7 days. So we were left with 1 in each cage with 1 sentinels and 1 principal infected animal. On day 21, we necropsied another animal, a principal infected animal, and then we used these 3 cats to reinfect them. And that was a question we had in fall of this year. Is somebody who is infected with SARS-CoV-2 and is recovered, is that person protected against the reinfection? Nobody knew it. So these studies we did in fall of last year, and I give you the summary in a couple of minutes. Which obviously it's important information which guides public health decisions and our decision makers. And these 3 reinfected cats, they were reinfected the same way we did the primary infection, and we euthanized them at 4 days post reinfection. So this is how we house these cats. We had to build extra cages in our BSL-3 Act. This is a facility. This is where you come in. This is a so-called bio bubble on the left, and then you come in, you wear peppers, respirators, you wear Tyvek suits, and in this case, when we deal with cats, we wear a polyester suit on top of our Tyvek suit to protect us. And we here are individual cages, because before you handle cats the way we do it, we have to sedate them, and that's what we do in these individual cat cages. And this is our treatment table, and we do a lot of environmental studies. And for example, we had air sampling in the air just to see where the virus moves, is there virus in the air, etc. So when we did the first study, we saw there is not much changes in body temperature, except maybe a spike on day 2. Red are infected and the sentinels are green. There seemed to be a spike in weight. There's not much, maybe a little drop on day 1, and then it comes back, so in an infected one. So there's not much of an effect on clinical symptoms. We also did all the blood. We looked for 14 different chemical biomarkers in the blood, and we did all the blood cells, etc. And we didn't see much except thrombocytopenia on day 5 after infection. Very important was, what about RNA and virus shed in various cavities? And we looked at the nasal cavity, oral cavity, and rectal cavity, and I will show you mainly now the nasal, upper respiratory, lower respiratory tract. So when we looked at nasal swabs, on day 0, there is nothing. This is red infected. On day 1 after infection, they already have a lot of viral RNA shedding, and it's maintained up to day 10. And then on day 14, these animals are negative and maintain this negativity to day 21. That's the day when we reinfected. If you look at the sentinels, which were added on day 1 to the infected animals already on day 3, the sentinels are positive, and they show a different kinetic of viral shedding from the upper respiratory, from the nasal cavities. When we did nasal washes, and we only did nasal washes on the animals we euthanized, we didn't do it on the animals we just swabbed and put back into their cages. We did it also only on these 2 animals on day 4, 7, and the 1 on day 21. You see, similarly as expected on day 4, you see a lot of virus shedding in the nasal washes, and on day 7 and nothing on day 21, which more or less similar data as with the nasal swabs. When we looked at BALF, and BALF is a bronchoalveolar lavage fluid. It's a fluid we get when we flush the lungs when we take the lungs out at necropsy, we flush them, and then we look in that medium which we use, we look for virus, and you see in the lung there is a decent amount on day 4 and day 7, but nothing on day 21. So they all seem to correlate with each other. And we did similar studies in the GI tract and in the oropharyngeal swabs, and they have similar kinetics as shown with the nasal swabs. And you can read that in the paper I refer to in a minute. We also looked in the upper respiratory tract and in the lower respiratory tract and the lymphatic tissues, as well as other organs. And as you see here, the upper respiratory tract, it's various tissues. You see a lot of RNA on day 4, 7, and maintain until day 21. That's the purple thing. And if we look in the lower respiratory tract, which is the lung, bronchi, most of these are negative on day 21. In the lymphatic tissues, they are maintained through all 3 days. And in certain organs, they are gone already at day 21, but others like the olfactory bulb, they are not. So we love, as I'm a pathologist, so I love pathology, and I go through that these are the bronchi. This is part of the respiratory tract. This is the mucosa on a normal animal. These are the bronchi on day 4, and you already see some infiltration here of neutrophils and so on, and monocytes. So there's already some minimal to mild infiltration. And if you look at day 7, it's even more here. And this is in the glands, the submucosal glands here. And if you look at day 21, it's negative. There's nothing pathological anymore. And we concluded you see an adenitis here on day 4 and day 7, but nothing on day 21 anymore. And if we look for the presence of RNA, that's what we use, that's what we call in situ hybridization. You see on day 4 and day 7, you see viral RNA into the tissue. And also if we use an antibody against antigen, this is called immunohistochemistry, you see that too, but nothing on day 21 anymore. So we concluded that we see significant histological changes in viral RNA and antigen at day 4 and 7, but not at day 21. The next question was antibodies, and we had these 4 animals which we had on day 7. The principal infected animals had neutralizing antibodies, and the sentinels became positive 3 days later. So the principal developed antibodies as well as the contact. So this is the summary of what I just said, and I go through that fast because it's published in this paper. What we saw most likely are changes which were due to virus infection, mainly in the upper respiratory and lymphatic tissues, and not so much in the lower respiratory tissue. And we did not see interstitial pneumonia as described in humans, but we saw efficient transmission to the sentinels. Then we did reinfection, and we used 2 independent experiments to study whether infected animals are protected against reinfection 21 days post-primary infection. And this is just to recapitulate these 2 experiments. We reinfected first these 3 cats. I described that before, and then we euthanized them 4 days later. And then we did a second study where we reinfected 5 cats. And we wanted to see, because in the first study we saw RNA shedding, we wanted to see whether these animals, which are reinfected and shed a little virus, can infect highly susceptible sentinels. And we euthanized these cats in the second study on day 4, 7, and then all the animals to see seroconversion on day 14 post-reinfection. And what did we see in terms of RNA shedding? These are infected, and these are sentinel animals in red and in blue. If you look, this is too much. If you look at the reinfected in green, you see shedding on day 1, and then it's gone and comes back on day 4. If we look, this is nasal swabs. If you look at a pharyngeal swab, we see similar. We see similar things, that you see shedding already early on. It goes away and comes back. So these reinfected animals seem to shed virus, but not these sentinels, which are kept in the same cage. We then looked at viral detection in nasal washes, upper respiratory, and in BALF. And this is a summary of that. On day 4, this is primary infected. You see a lot of virus on day 4, but nothing on day 21 at the time of reinfection. On day 4 after reinfection, there's not much virus in there, only 1 out of 4 or 5 animals. However, on day 7, both animals we tested were positive. And then the virus disappears by day 14. In the lung, the situation is different. On the primary infected animals on day 4 have a lot of virus in the lung, but nothing on day 21. And if you reinfect these animals on day 4, 7, and 14, there's nothing in the lung, indicating that there's not much immunity in the upper respiratory tract, but a lot of immunity in the lower respiratory tract in the lung. And that's very important because disease, if you have lung disease, you get really sick. If you have just upper respiratory disease, you get a cold, or you get a running nose, etc. It's not so critical. We looked for virus neutralizing antibody, whether there's an anamnestic immune response, meaning that the virus replicates and you get higher antibody titers if that happens. And this is our first group. These were infected on day 21 and euthanized on day 4 after reinfection, so they were reinfected. There is no difference in antibody titers. But if we look at the next pair, which was reinfected on day 21 and then euthanized on day 7, you see already a difference in antibody titers. And this is also true for that one animal, which was reinfected on day 21 and euthanized on day 14. None of our sentinel animals, however, seroconverted. So there was no transmission to sentinels. So we concluded that, I went through all this, we saw limited viral shedding from nasal and oral cavities, and we saw some, I didn't have time to that, some pathological changes. However, no viral antigen or RNA in the tissues. We saw an anamnestic immune response after reinfection, which indicates that some virus, most likely in the upper respiratory tract, replicated and induced some antibody responses. But we did not see any shedding of virus to the sentinels, and they did not seroconvert. So our final conclusions are cats are highly susceptible to SARS-CoV-2. They respond in our settings asymptomatic, no clinical signs, but they efficiently transmit it to other cats. And that's the question, they most likely can also transmit it to other animals, including humans. And the other conclusion is that if you are infected, at least cats are infected and recover from infection, that these immune responses provide partial immunity against reinfection, which is non-sterilizing. You get really good protection in the lower respiratory tract, but not in the upper. So I go on to another study, which is not published yet, and I have 4 or 5 slides on this. We also studied white-tailed deer. I do a lot of work with wildlife, and so we decided to do white-tailed deer for various reasons. Why? White-tailed deer are the most widely distributed wild ungulates in the Americas. We have 30 million in the US. Many cervid farms exist which house and breed white-tailed deer in the US, and white-tailed deer are also present in other countries around the world. And this is how they look. And they're not very easy to handle. That's why we have very different housing and penning compared to other animals. This is a BSL-2 level. They still have bedding. If they go into the high containment facility, they have the same pens, but no bedding anymore. They have special floors where they live on. So what was the evidence that white-tailed deer are susceptible? Again, these studies, these structural studies I described, were done by many people. And one study described that deer ACE2, so the receptor in deer, would easily, with high efficiency, bind the spike protein. And this is the study by Damas et al. And he or she showed that white-tailed deer shown here have a high potential to get infected, also reindeer. So what we did is very simple experiment, because you cannot have a lot of animals. The whitetail need more space, etc. And in the biocontainment, we only had 6 animals, 4 females, which we inoculated as with the virus, and we had 2 sentinels, also female. And we did daily observation, temperature, not easy. We did the swabs, similar to what we did with the cats, and we tested that at up to 18 days. And then we did postmortem examination on day 4 and day 18. And we infected these animals. We changed it a little bit. We infected them again with 1 million virus, but we not only used the strain from Washington State, the first one isolated in the United States, we also used the variant of concern. And you all hear about alpha, beta, gamma, delta. This was an experiment done in February this year. We used an alpha variant of concern to see which virus works better and transmits better. And we did intranasal and per os inoculation simultaneously, did the whole virology and serology. So this is how we do this study. You inoculate these animals intranasal per os with these 2 viruses. A day later, you add these sentinel animals, and then we swap them at various days, 1, 4, and so on, and do fecal samples, blood collection, etc. You necropsy, euthanize 2 deer on day 4, and the rest we euthanized on day 18. What did we see? To our surprise, all 4 principal infected animals already on day 1 had virus coming out of their nose. Only 3 out of 4 from their oropharyngeal. And later on, we found 1 who had also rectal positivity. And everyone was negative from the principal animals on day 10. So nasal swabs are the best swabs. We only had 2 left here, were positive still. If we looked at the sentinels, they were negative on day 0. We co-habitated, commingled them 2 hours after. We swapped them, sorry. We cohabitated them, commingled them 1 day after infection and swapped them 2 hours later. And in 2 animals, in one of these animals, we already found 1 out of 2 PCR reactions suspect. So, but on day 3, it's very clear. Both of these sentinels were positive by nasal swabs, and 1 out of 2 by oropharyngeal, and 1 of them also became positive later on rectal swabs, and they were all negative again. Actually, not all. One was still positive on day 10 here, but all negative on day 14. So this is how the next question for us was, if we have these sentinels here and the principal infected here, what kind of viruses do these principal infected animals, these 4, shed? So they were inoculated with 42% of the ancestral virus and 58% of the alpha variant of concern. The sentinels got nothing. They were only co-housed after 1 day. So this was the inoculum. On day 1, obviously there was nothing in the sentinel, but 2 out of these 4 animals already had 100% of the B117, which is the alpha variant of concern. And 2 still had the ancestral virus as a percentage, significant percentage. On day 3, all of these 4 principal infected animals had only the alpha variant and not the ancestral virus anymore. And on day 5, we could isolate out of these sentinels both only the alpha variant, but none of the ancestral. So we did also histopathology. I love that. On the left, the trachea, in the middle, a bronchus, and here on the right is a bronchiole. And I want to show you here, this is RNA, beautiful in the trachea. There's the sledding of cells, and you see the viral RNA in there. This is a bronchi. You see here, beautiful also viral RNA, and there is a bronchitis, inflammation of cells. And if you look in the bronchioles, you have also inflammation around the bronchioles, but we couldn't find, for whatever reason, RNA in this sample. So we also did antibodies, and when you see here on day 7, these animals already, the principal infected animals had high antibody titers on day 7. On day 14, both the principal infected and the sentinels had antibodies. Same we do with ELISA. But to summarize this part is we had white-tailed deer, and we first did white-tailed deer cell lines, and they were very easily infectable primary deer cell lines. And that was the first study we did, and then we did the transmission in real white-tailed deer. We could show that they're highly susceptible to SARS-CoV-2, that these adult white-tailed deer transmit SARS-CoV-2 easy to Sentinel. We showed that the alpha variant of concern replicates and transmits much more efficiently than the ancestral USA strain. And all of these animals, whether they were principal or sentinel, seroconverted. And we could also show that our assays work very well. There was another paper published a couple months ago by Palmer and others, and they did white-tailed deer fawns, young animals, 3, 4, 5-month-old animals, and they showed a similar in susceptibility rate as we showed here. I think I'm at the end of my time, so we cannot go through the therapeutic approaches anymore. We did a study in hamsters which we just published in Cell, and I have a disclosure here. We have a patent submitted under therapeutic treatment, and I just go to the summary here. We have a system to do antiviral testing in cell culture, and we used a repurposed library called Reframe Library from Scripps. And for example, if you have remdesivir, you see here no drug treatment, positive cells. If you have drug treatment, the cells are not so negative or very good. And we identified with our collaborators about 30 promising antivirals we are testing right now in different, especially first in hamsters, and then we go in on human primates, and then in clinical trials. We did remdesivir and topotecan. Topotecan is a topoisomerase inhibitor, a cancer drug, which is FDA approved and used in hamster model. And I want to go fast through this and go through the summary. Here it is. When we treat SARS-CoV-2 infected hamsters with topotecan alone post-infection, so not prophylactic, but therapeutically, we see limited lung inflammation, but we do not suppress viral replication and virus dissemination. So we need, obviously, an antiviral treatment in order to help the patient. So we combined, if we do remdesivir alone, which is an antiviral, which is approved by the FDA for treatment of patients, if you do it alone, it limits, and before infection, it limits virus shedding, but not this inflammation we limit with topotecan. And if we combine topotecan with remdesivir, we see we can control virus replication. At the same time, we limit lung inflammation. And as you know, this cytokine storm is what kills people. It's not more or less the virus replication. It's more the immune reaction to the virus replication, which is very dangerous and can really lead to death. And so we believe that this is a treatment, an interesting treatment for COVID patients, and we are doing that right now. We got permits to study the safety and efficacy of this strategy in clinical trials. We are doing it in India right now. And the paper where this is published is a Cell paper, just recently came out by Ho and co-workers. So this is what we have done in the last 18 months. We have completed studies in pigs, cats, hamsters, that's what I discussed with you today, and also white-tailed deer. We have also completed studies in sheep, I didn't have time, and in ferrets. And we were asked to also study dogs because this is an important other companion animals which has shown a high susceptibility or medium susceptibility to SARS-CoV-2. I'm at the end, and I mentioned my collaborators before, and now I'm really ready for questions if you may have any questions for me. Thank you for your attention.